Hello everyone, as we already covered prokaryotic cell, here we are going to cover about eukaryotic cell in detail. Coming to the etiology of the eukaryotic cell, it is Greek word where eu means true, karyon means nuclear, where it contains true nucleus and membrane-bound organelles like endoplasmic reticulum, mitochondria, vacuole, lysosomes, Golgi apparatus, etc. It also contains complex locomotory and cytoskeletal structures which helps in locomotion, and these provide strong structural support to the cells. The genetic material is organized into chromosomes where the nucleus is covered by nuclear membrane. Coming to the examples of the eukaryotic cells, these are examples: protista, fungi, plant and animal cells. As we all know, eukaryotic cells are not identical. Let's differentiate between plant cell and animal cell. Here, plant cell contains cell wall as outermost protective layer, which is made up of cellulose. These plant cell surrounds the plasma membrane, where in animal cell the cell wall is absent. The outermost covering is the plasma membrane. Coming to the size of the cell, plant cell is larger than the animal cell. In mature plant cell, it contains large central vacuole which occupies up to 90% volume of the cell. Coming to the plant cell, animal cell, here the vacuoles are small, many and temporary. Coming to the plant cell, here the plastids are present which involved in photosynthesis. The plastids are absent in the animal cell. The centrioles and centrosomes are present in the animal cell and absent in the plant cell. Let's discuss about structure of plasma membrane, which is also known as cell membrane. As we all know, it is a selectively permeable in nature. It regulates the movement of substances in and out and interacts with the outside world. The structure of plasma membrane is studied on the human red blood cell, and detailed structure of the plasma membrane is studied after the invention of electron microscope in 1950s. Coming to the composition of the plasma membrane, it contains lipids, proteins and carbohydrates. Coming to the lipids, these are 40%, proteins which are 52%, and it also contains carbohydrates on the cell membrane which is very less amount. Coming to the lipids, these also contains phospholipids and cholesterol. The phospholipids which has hydrophilic polar end towards the outside the membrane, which contains saturated hydrocarbons, and it also contains non-volatile towards inner side, which is hydrophobic in nature. Coming to the proteins, on ease of extraction, these are different classified into two types: integral proteins and peripheral proteins. Integral proteins which are partially or totally buried in the membrane. These are also known as transmembrane proteins because these proteins which are present throughout the lipid bilayer. Coming to the peripheral proteins which lie on the surface of the plasma membrane. Carbohydrates, as we already discussed, these are present in very less amount. The most improved model of plasma membrane, which is known as fluid mosaic model. This is proposed by scientists Singer and Nicholson in 1972. According to this model, they said that the lateral movements of proteins within the lipid bilayer is due to quasi-fluid nature of lipids, and this fluid nature of lipids which helps in growth of cell division, endocytosis and secretions, etc. And this ability to move within the membrane is measured as its fluidity. These are the various cell organelles that are present in the eukaryotic cell, like nucleus, lysosome, ribosomes, endoplasmic reticulum, mitochondria, Golgi apparatus, etc. Let's discuss structure of each and every cell organelle and functions performed by them in detail. Coming to the endomembrane system, it composed of group of membrane-bound organelles like endoplasmic reticulum, Golgi complex, lysosome, vacuoles, which are structurally different but functions in a coordinator manner. The mitochondria, chloroplast which is present in plants, and peroxisomes, these are not the components of endomembrane system, as these are not functions in a coordinated manner. Coming to the endoplasmic reticulum, these are the tubular structures which are scattered in the cytoplasm. Let's discuss the structure of endoplasmic reticulum in detail with the help of diagram. Here, the endoplasmic reticulum, it divides its intercellular space into two compartments, which are luminal compartment, which is present inside the endoplasmic reticulum, and extraluminal compartment, which is present towards the cytoplasm. Again, based on presence of ribosomes, these endoplasmic reticulum is divides into two types. Those that pierce ribosomes on its surface are known as rough endoplasmic reticulum, which involved in the synthesis of proteins, and these rough endoplasmic reticulum, which continues with the outer membrane of nucleus. Coming to the smooth endoplasmic reticulum, these do not contain any ribosomes on its surface, and these involved in the synthesis of lipids, and these also synthesized lipid-like steroid hormones. Let's discuss about Golgi operators in detail. These are densely stained reticular structures which are present near the nucleus of the cell. These are first observed by Camillo Golgi in 1898. Coming to the structure of the Golgi operators, it consists of four parts, which are cisternae, which has scissor forming phase, and tubules, vesicles, and Golgian vacuoles. Let's discuss structure of each part in detail. Coming to the cisternae, these are flattened sac-like structures which has scissor forming phase and maturing phase. As these two phases different to each other, these are interconnected in its function. These cisternae are parallel to each other and arranged concentrically near the nucleus with diameter 0.5 micrometers to 1 micrometer. Coming to the tubules, these are flattened round structures which are arises at the periphery of the cisternae. Vesicles, these are small round structures which are arises at the edges of the cisternae. Coming to the Golgian vacuoles, these are large spherical structures of the Golgi operators. Let's discuss functions of the Golgi operators. These helps in processing, packaging, and transporting the materials to the intracellular targets or outside the cell. The proteins which are synthesized in the endoplasmic reticulum are transported towards the cisternae of the Golgi operators and fused to the cis phase of the Golgi operators and moves towards trans phase and maturing phase of the Golgi operators, where these proteins are modified before they are releasing out of the trans phase, and these are later acts as lysosomes and perform various functions in the cell. Let's discuss the structure of lysosome in detail. These are tiny spherical sac-like membrane-bound vesicular structures which are formed by packing in the Golgi operators. These are rich in hydrolytic enzymes like hydrolysis, which are lipases, carbohydrates, proteases, and nucleases. These are active at acidic pH and helps in capable of digesting carbohydrates, proteins, lipids, and nucleic acids. This also contains transport proteins on the surface of them. Let's discuss about mitochondria. As we all know, this is cylindrical, a sausage-shaped double membrane organelle which is present in the cytoplasm of the cell, which has 0.2 to 1 micrometer in diameter and on an average, which is 0.5 micrometers, coming to the length of the mitochondria, which is 1 to 4.1 micrometer in length. And to see the mitochondria under the microscope, we have to use special stain known as Jonas green B. There are variable number of mitochondria are present in the cell, different types of the cells depending on the physiological activity, shape, size and energy requirements of the cell. Let's discuss structure of mitochondria in detail with the help of diagram. Coming to the structure, it is double membrane bound organelle which contains two membranes, that is outer membrane which continues as limiting boundary of the mitochondria and inner membrane which forms number of infoldings known as cristae, and the lumen of mitochondria again divides into two compartments. Outer compartment which is present between the outer and inner membrane of the mitochondria, which is known as intermembrane space or perimitochondrial space. Coming to the inner compartment which contains dense homogeneous substance known as matrix. These contains circular double-stranded DNA molecules, RNA molecules, ribosomes which are 70S, and these also contains components required for the protein synthesis. Coming to the functions of mitochondria, these are the sites of aerobic respiration and these are also known as powerhouses of the cells, as these contains enzymes required for the synthesis of ATP, and these produces energy required by the cell to perform various functions. There are two membranes in the mitochondria. The two membranes have its own specific enzymes which are associated with the mitochondrial function. Let's discuss about ribosomes. These are observed by George Pellet in 1953 as dense particles. These are non-membrane bound organelles which are present both in prokaryotic and eukaryotic cell as 70S and 80S ribosomes. It has two subunits, larger subunit and smaller subunit, which is 60S and 40S in the eukaryotic cell. Here in prokaryotic cell 50S and 30S, this is the larger subunit and this is the smaller subunit. Here S stands for Svedberg unit, which is segmentation coefficient. Here it measures the segmentation rate. Coming to the structure of the ribosomes and its functions, the ribosomes which helps in synthesis of proteins and translating mRNA into specific sequences of amino acids. Let's discuss about cytoskeleton. These are elaborate network of filamentous proteinaceous structures like microtubules, microfilaments and intermediate filaments which are found in the cytoplasm of the cell and helps in the providing strong structural support to the cell movement of the cell and maintains the shape of the cell. Let's discuss with the help of diagram. These are microtubules which are hollow cylindrical structures found in the cytoplasm of the cell and made up of tubulin protein and which forms the cytoskeleton of cilia and flagella. These are microfilaments. These are unbranched structures found in the cytoplasm of the cell and composed of actin and myosin proteins, helps in the cell division by forming an furrow. These are intermediate filaments. These are hollow filamentous structures helps in the forming scaffolds for chromatin material. Let's discuss about structure of centrosomes and centrioles. Here the centrosome consists of two cylindrical structures which lie perpendicular to each other and has an cartwheel-like organization are known as centrioles. These are surrounded by amorphous pericentriolar materials. Let's discuss structure of centriole in detail. Coming to the structure of centriole, it has 9 evenly spaced peripheral fibrils which are known as triplet fibrils made up of tubulin protein, and these triplets are linked with adjacent triplets with the help of AC linkers. and it also has central proteinaceous hub which are connected to these triplets with the help of radial spokes. These centrioles forms the basal body of cilia and flagella, and spindle fibers gives rise to spindle apparatus during the cell division. Let's discuss cilia and flagella. These are hair-like outgrowths of cell membrane. Coming to the cilia, these are small structures which helps in the movement of cell and surrounding fluid. Here, the flagella, which is longer one, which is responsible for cell movement. Structurally, the prokaryotic flagella is different from the eukaryotic flagella. Here, it consists of basal body, rootlets, basal plate, shaft, and axoneme. Let's discuss it with the help of diagram. Let's see the structure of flagella. It's an axoneme, a shaft of the flagella. It contains 9 microtubules with 2 central tubules which are connected with bridges and enclosed by central sheath. So these flagella has an arrangement of 9 plus 2, where 9 means it has 9 microtubule doublets and 2 means 2 central tubules. And both cilia and flagella emerge from the centriole-like structures called basal bodies. As we already differentiated between plant cell and animal cell, let's discuss some of the organelles which are present only in plant cells like plastids and outermost covering of the plant cell, which is cell wall. Coming to the plastids, these are found in all plant cells and euglenoids. Depending on its color and types of pigments present in them, these are divided into 3 types: leucoplast, chromoplast, and chloroplast. Coming to the leucoplast, these are colorless plastids which are present in non-green cells. These are of 3 types: amyloplast, which contains starch, example potato tuber; alleloplast, which stores oils and fats; alluroplast, which stores proteins in them. Coming to the chloroplast, these chromoplasts, which consists of fat-soluble carotenoid pigments like keratin, xanthophyll, etc. These produces colors like yellow, orange or reddish orange to the ripening fruits, because here the chloroplasts are transformed into chromoplasts, which gives colors to the ripening fruit. Coming to the chloroplast, these are lens-shaped oval or spherical ribbon-like structures which are present in the mesophyll cells of the leaves. These are greenish plastids which are present in green cells of the leaves. These contain chlorophyll and carotenoid pigments which are responsible for trapping light energy, which is essential for performing photosynthesis. These are about 5 to 10 micrometer in length, 2 to 4 micrometer in width. There are variable number of chloroplasts are present in cell, like in Chlamydomonas, which are present 1 per cell, and in green algae, these are present 20 to 40 per cell. Let's discuss structure of chloroplast with the help of diagram. Here, the chloroplast is double membrane organelle which consists of 2 membranes, which is outer membrane and inner membrane. Between outer and inner membrane, it consists of space known as intermembrane space. Inside the inner membrane, the fluid-filled space known as stroma, which contains number of flattened membranous sacs known as thylakoids, and these thylakoids are arranged in stack-like piles of coins known as grana, singular, which is known as granum, and these grana are also known as integral thylakoids. And thylakoids of different grana are connected with help of tubules known as stoma lamellae. These consist of 70S ribosomes, which are smaller than the cytoplasmic ribosomes. And these chloroplasts, as we already discussed, these consist of pigments like chlorophyll, which trap light energy essential for photosynthesis. These perform photosynthesis, light reaction in the thylakoids and dark reaction in the stroma. These also contain starch in them. These mitochondria and chloroplasts are semi-autonomous organelles because they are capable of self-duplication, as they have their own nucleic acid like double-stranded DNA and RNA molecules, 70S ribosomes, and it also has ETS and ATP forming machinery in it. Let's discuss about cell wall. It is non-living rigid structure which is found in fungi, algae and plant cell. Here in fungi it is composed of chitin, in algae it is composed of cellulose, galactans and some of the minerals like calcium carbonate, and in plant cell it is composed of cellulose, hemicellulose, pectin and proteins. Based on age of the plant cell, in young plant cell it forms a single layer which is thin, elastic and capable of growth, which is known as a primary cell wall, and in mature plant cells inside to the primary cell wall it forms as secondary cell wall. Coming to the middle lamella, it is the connecting material which holds the adjacent cells together, as you can see in the picture. Here, the cell wall and middle lamella are transversed by plasmodesmata, which means these plasmodesmata which extends or passes across the cell wall and middle lamella and connects the cytoplasm of neighboring cells, and this allows transport of water, nutrients and provides cell to cell communication between the plant tissues. Coming to the functions of the cell wall, it maintains shape of cells and protects cell from mechanical damage and infection, and it provides cell to cell interaction, and it provides barrier for undesirable micromolecules. Let's discuss about vacuole. It is present in both plant and animal cells, but in plant cell it occupies large volume up to 90% volume of the cell. Let's discuss structure of vacuole. It is single membrane bound organelle in the cytoplasm. It contains water, sap, excretory products and waste materials. The single membrane, which is known as tonoplast, it facilitates transport of number of ions and materials against the concentration gradient into the vacuole. There are many types of vacuoles like contractile vacuole, which is found in amoeba. This helps in osmoregulation and excretion, and foot vacuole, which is present in protists, which helps in engulfing the food particles, and gas vacuole, as we already discussed about it in prokaryotic cell. These are found in prokaryotes, which provides buoyancy to the cells. As we already discussed about various cell organelles of eukaryotic cell, its structure and function in detail, here we are going to cover about nucleus, which is the largest cell organelle and controls all the activities of the cell. It was first described by scientist Robert Brown in the year 1831, and based on number of nucleus present in a cell, if the cell contains two nuclei, then it is known as binucleate cell, as in case of paramecium. If cell contains many nuclei, then it is known as multinucleate cell, and if cell lack nucleus, as in case of mature mammalian RPC and sieve-tube cells of vascular plants, then it is known as anucleate cell. Let's discuss about structure of nucleus in detail. At non-dividing phase of the cell, which is known as interphase nucleus, has nucleolus, chromatin, nuclear membrane, perinuclear space, and nuclear pore. Let's discuss about each and every part of the nucleus in detail. Coming to the nuclear envelope, which is the membrane that separates nucleus from the cytoplasm, is differentiated into outer membrane, which continues with the rough endoplasmic reticulum, and inner membrane, which is smooth. Between these two membranes, there is a space which is known as perinuclear space, and there are nuclear pores which are formed by fission of these two membranes, and this forms as passage of substances to move inside and outside the cell. And there is transparent fluid which fills the nucleus, which is known as nucleoplasm, that contains nucleolus and chromatin material. Coming to the nucleolus, this is non-membranous spherical structure. This is a site for ribosomal RNA synthesis, and there are large number of nucleolus present in a cell, and that indicates that this cell is involved in protein synthesis. If there are large number of nucleolus in a cell, that indicates the cell is actively involved in protein synthesis. Coming to the chromatin material, it's a Greek word where chroma means color. It is the highly extended and elaborate nucleoprotein fibers. Here, it is named by scientist Fleming based on its ability to stain with some of the basic dyes. And this chromatin material, which is composed of DNA molecules, some of the basic proteins like histones, non-histone proteins, and RNA molecules. In human cell, the DNA, which is 2 meters long thread-like structure, which is distributed among 23 pairs of chromosomes. At different stages of cell division, where we are going to discuss in chapter 10 cell cycle and cell division in detail, the chromatin material is condensed to form chromosomes. Let's discuss about structure of chromosome in detail. Here, the chromosome consists of two identical halves which are known as chromatids, and these chromatids held together at a point which is known as centromere, and this centromere appears as narrow region which is known as primary constriction, and sides of this centromere there is a disc-shaped structures which are known as kinetocodes. Based on position of this centromere, there are 4 types of chromosomes. Let's discuss about each and every type of chromosome in detail. Metacentric chromosomes, if the centromere is present at the center of the chromosome and divides into 2 equal arms. If the centromere is present slightly away from the center of the chromosome, then it divides the chromosome into one shorter and one longer arm. Then it is known as submetacentric chromosome. If the centromere is situated close to its end and divides into one extremely short and one extremely long arm, then it is known as acrocentric chromosome. If the centromere is present at terminal portion, then it appears as single arm. It is known as telocentric chromosome. And few chromosomes has additional constrictions at its end, which is known as secondary constrictions, and which is present beyond to the secondary constrictions as small fragments. These are known as satellites. If you like this video, don't forget to like, share and subscribe to my YouTube channel. We'll meet again in our next video.